Hello, this is Gina Piscatelli with a lecture for Anatomy and Physiology 1 at Madison Area Technical College. In this lecture, I will talk about the cell cycle and mitosis. Just so that you're aware, there are a number of good videos that talk about this topic, and they're listed on this slide. I want to let you know that you'll be given these links through Blackboard, but in addition, you'll be given this PowerPoint, and if you open this PowerPoint in presentation mode and click on any of these three links, it'll take you directly to these videos. So these are for extra tutoring in cell division and the cell cycle, and they're very good. Before I get started about talking about the cell cycle or mitosis, there's a few terms I need to introduce. First is the term somatic cell. In this lecture, we will be talking only about somatic cells, and you need to know what they are. This term somatic is a general term for a typical body cell, meaning that it won't develop into a reproductive cell known as a sperm or an egg. So we'll only talk about stages that a somatic cell goes through. Now, the life cycle of a cell is a phrase that I kind of coined, I guess. It's the stages of activity that a somatic cell can display during its lifetime. What does it go through from when it forms until it dies? And there's two different stages that we will talk about. One is the quiescent stage, and the other is the dividing stage. So a quiescent cell is a cell that's in quiescence. And what that means is that it's metabolically active. It is functioning in the role that it has in the body, be it a bone cell or a skin cell or a muscle cell. But it is not preparing to divide in any way. It's just busy doing its job. Whereas a dividing cell could be in one of two different phases. It could be preparing to divide or maybe actively physically dividing. So we'll look at the difference between those two things in a little bit. In the field of cellular biology, there's a term, this last term, called the cell cycle. This term was defined to mean the stages through which a cell goes that's going to divide. The stages of a dividing somatic cell. Now, that's preparing to divide as well as actively dividing. So part of the cell cycle will be the physical division, right? And one area that you might have heard of already for cell division is studied called mitosis. That is just part of the stages of the cell cycle. Technically, mitosis is just the process by which the nucleus divides. Chromosomes have to move and be distributed to new daughter cells. Now, these chromosomes during mitosis can be seen with a microscope. And so these stages of a dividing cell were seen first. They could be seen with a microscope versus any molecular means or chemical means. So the cell cycle refers to a dividing cell only. Here are the stages of the life cycle of a somatic cell. What are the things that a cell goes through during its lifetime from birth to death? Well, after it's initially formed, it's in quiescence. It's a quiescent cell, and it's actively just doing what it's supposed to do. It's a bone cell, a skin cell, whatever. If it receives a signal to divide, and that's usually a chemical signal, then it will enter the cell cycle, of which mitosis is a part. So during mitosis, the nucleus will divide into two. the end result of cell division is two new daughter cells. Now these two daughter cells most likely will not begin dividing right away. They'll grow and then they will perform the role in the body that they need to. They've been told to be, like a bone cell, a skin cell, a muscle cell. And so they enter a stage also called quiescence because they're metabolically active but not dividing. Eventually, a quiescent cell, or a cell that's not dividing, ages. It goes through senescence or enters senescence, an aging process. And that's because organelles like mitochondria begin to become fragile and degenerate. And eventually the cell cannot function as well. And the phospholipid bilayers may break apart, etc. So when a senescent cell eventually degrades and dies by this process, it's called necrosis. There's another process of cellular death, which is called apoptosis. And that's programmed cell death involving a chemical signal in which a cell is basically told to go through a series of steps to divide. We aren't going to talk about the difference that much, but I do have the differences outlined on the next slide. So you'll be able to see that, you'll be able to see the definitions of each of these. So quiescent cell is the stage when a cell is metabolically active but not dividing. Cell division is the process of one cell dividing into two daughter cells, and we can call that the cell cycle. Senescence is aging, and cell death can happen by either apoptosis or necrosis. Now, what are the purposes of cell division? Well, in the body, from birth until death, there are various times when the amount of tissue we have needs to change, particularly grow. So when cell division happens, as is shown in the box on the right, you can see that if you start out with a certain number of cells and then cell division happens, you have more cells, and this increases. This is the way our body develops, and you can see in this example I'm showing you how a particular gland developed. Another reason is for cell division to occur is to replace damaged cells. And an example of this would be when you are cut and that cut heals. So there are lost cells, a depletion in the cell number because they've been destroyed by the cut. But then the tissue repair happens by the process of cell division. Healthy cells divide and replace the damaged cells. The cell cycle, as I mentioned, is a process of a dividing cell, what a dividing cell goes through, and it is very tightly regulated. There are phases for preparing to divide, and there are phases of actual physical division. So the phases of preparing to divide include growing, replicating DNA, which means copying the DNA, and replicating organelles, which means copying organelles, increasing the number of organelles. And these phases have symbols that we use. G1 is called gap 1, and it's the cell growth phase. The S phase stands for DNA synthesis, so that's when DNA is being copied, so there'll be enough for 2 cells. And the G2 phase, also called the gap 2 phase, is when organelles are replicating, particularly mitochondria and centrioles. Now, the phases of division include nuclear division and cytoplasmic division, which will include the organelles. Nuclear division can also be called mitosis, and cytoplasmic division can be called cytokinesis. Both of these happen in a certain phase called the M phase of the cell cycle. So there's been a lot of terms thrown at you. I'm going to just show you a picture of the cell cycle. Hopefully this makes more sense to you. So this cycle or circle represents different phases that a cell goes through when it has already received a signal to divide. So it is entering what is known as the cell cycle. It first enters this green phase known as G1. This is when cell growth happens. Then it enters the S phase, which is when the DNA is copied or replicated. Then it enters G2, this blue phase, and that's when organelles replicate, particularly the centrioles, which are needed for the next phase, which is called the M phase, and that is named for mitosis primarily. So M phase is when the nucleus divides and the cytoplasm divides. It's just that there's two terms subdividing the M phase. So initially what happens before a cell enters the cell cycle is that you have a quiescent cell that receives some sort of signal to divide, and then it has to enter the cell cycle. And it always enters at G1. Now, the quiescent cell, because of this cell cycle nomenclature, has also been given a symbol, and that's G0 or G0. So this is the quiescent cell here in the box. It's a cell that's going to enter G1 and go through stages. And we're going to talk about what happens in each one of those stages next. So in G1, because protein synthesis is happening, the cell enlarges. Many proteins are going to be needed, including enzymes that will facilitate the process in S phase, G2 phase, and M phase. In the S phase, DNA is replicated. Now this bears a little scrutiny. DNA replication means that all 46 chromosomes are copied so that you then have 46 to go into each of two cells. There's an enzyme that copies DNA. It's called DNA polymerase. And what happens is the DNA helix unwinds. So these two pink strands of the DNA helix in a chromosome will split apart. And each strand will become the template for a complementary strand. So the complementary strand that was created by DNA polymerase is shown as blue. Just to give you an idea, imagine that in this original chromosome there was a C nucleotide here and a G nucleotide here, and they were bound to one another. Okay, so whoops, oh, I didn't mean to do that. Okay, the picture's back at a normal size now, and what we have are C and G bound to one another. Now DNA polymerase will synthesize a new strand for each one of these, and the new strand will be complementary to the original strand. So, for example, if here was a G, the new complementary strand is going to have a C here. And over here, where there was a C, there will be a G complementary to it, synthesized along the length of the strand. So what you started with is identical to the two products you end up with. So that's how DNA replication occurs. I do have a better picture for you though. So first I should tell you that the DNA exists in a form called chromatin, and that means that it's loosely coiled. It's not tightly coiled, and that is so DNA polymerase has access to this base sequence on the interior of the chromosome. If it were tightly wound up, DNA polymerase wouldn't fit in there. So this is a better picture for DNA replication. This is happening during the S phase, so we don't get lost. This is our double helix. Both strands are shown in navy blue. And notice how they split apart. And there's a DNA polymerase that works on one strand, and there's a DNA polymerase that works on the other strand. And they both are synthesizing a new complementary strand to the original strand. And the new strand is shown in red. So before the S phase, each chromosome existed as a loosely coiled single structure. Even if it's in a hairpin loop like this, it's linear. It's just like, one double helix. But after the S phase, there are now two copies, I guess, of the chromosomes that are attached to one another. And where they're attached, that point is called the centromere. And at this point, we still call this one chromosome, which is very annoying to students because it looks like two. But each of the strands, I guess DNA helices that have been formed, are called sister chromatids. So before the S phase, the chromosome is a single double helix, one double helix. After the S phase, there's two double helices joined by the centromere, and each double helix is called a sister chromatid. Now, after the S phase occurs, there is a set of enzymes that check to make sure that the DNA has been copied correctly. So there's a checkpoint here. If, for some reason, the DNA has not been copied or replicated correctly, the cell will exit the cell cycle and undergo apoptosis, or programmed cell death. But we're going to assume everything went fine and that the cell enters the G2 phase. During this phase, as I mentioned, organelles replicate, and the organelles that I've drawn on this cellular picture are the centrioles. The centrioles are yellow tube-like structures, but you already know that they're made up of microtubules. So there's always two in a cell that's not dividing. When the centrioles replicate, now there's four. And so two of them migrate to each end of a cell. We call it opposite poles of a cell. And in addition, these centrioles begin forming additional microtubules. And these microtubules arrange to form what we call a spindle apparatus. This spindle apparatus is important for moving the chromosomes around during nuclear division. During this time, the DNA is still in a form of chromatin, loosely coiled. You cannot see it. It's not condensed. So these so far are the phases that we've talked about in a dividing cell. G1 is when cell growth happens. The S phase is when DNA synthesis happens. The G2 phase is when the centrioles replicate and the spindle apparatus forms. Now we're going to enter this final stage of the cell cycle. And this is called the M phase. And two things are going to happen: mitosis and cytokinesis. So during the M phase, mitosis involves nuclear division, which the most important part of that is to make sure that the chromosomes are split and evenly distributed into two daughter cells. And the cytoplasm has to divide as well. So we start off with a cell that looks pretty normal. Its centrioles haven't actually migrated yet in this picture, but one will go over here. So this is a cell before it's actually, done any of the cell cycle phases. And in the end result, we should get two daughter cells that only have two centrioles, but the proper number of chromosomes. We want to make sure that each daughter cell has the right number. Okay, so one thing I have to throw in here is that G1, S, and G2 historically weren't even known about. We didn't even know that all those things were happening. We just saw under the microscope a cell that you couldn't see the chromosomes. And so you could not tell that a cell was dividing. So scientists were really only studying cell division with a light microscope. Therefore, they could only see chromosomes in cells that are dividing. They didn't know anything about the S phase, G1, G2, they had no idea. So they concluded that if you couldn't see the chromosomes, the cell was not dividing. Therefore, they had this term for a non-dividing cell called interphase. That means you can't see the chromosomes. And that now we know that interphase has all these subdivisions, part of the cell cycle. So it is not possible to distinguish, at least with a light microscope, a cell that is intending to divide but still in interphase, G1S or G2, versus a cell that isn't even intending to divide. They look identical. You cannot tell the difference because it's happening on a molecular level, at least not with a light microscope. So these are the phases of mitosis or nuclear division. That's what we're going to talk about first. And in nuclear division, this interphase stage doesn't really count because you can't see anything in the nucleus. So the onset of mitosis is when you can first see chromosomes condensing into these linear strip thread-like things. So that's shown in this diagram where you can begin to see the chromosomes, and in this second picture most clearly. Early prophase, you can kind of tell that there's something happening in the nucleus, but the chromosomes are not tightly coiled and condensed yet. The next thing that the cell does is, because of the spindle apparatus's microtubules, the chromosomes are lined up at the equator of the cell. It's called the metaphase plate, actually, not the equator. And this is known as metaphase. So now the chromosomes are all lined up in the central part of the cell. Then the centromeres split apart, and each sister chromatid is pulled towards the opposite end of the cell. So in anaphase, you can see it probably most clearly here. You can see that individual chromatids are being pulled apart. So the centromere has split. And this picture also shows anaphase, where the chromosomes are being split apart. The chromosomes are shown in blue, of course. And now finally what has to happen is that a nucleus has to form around the individual chromosomes. And this occurs in the final stage of mitosis called telophase. Finally, a cell membrane is going to form, and that's part of cytokinesis. So let's see if we can put this all together. In interphase, you can identify a cell that's in interphase because you see an intact nuclear membrane. You can also see a nucleolus because RNA is still being made. The chromosomes are not visible you cannot see chromosomes in an interphase cell. You don't know if the cell is in G0, G1, S, or G2 unless you see there are 4 centrioles. Now you know that this cell has completed G2, but otherwise you wouldn't know. And truthfully, you cannot see those centrioles with a light microscope anyway. So most of the time you can't tell, you just say interphase. I don't know, it's in interphase. So the onset of mitosis, or the M phase, is prophase, and that is when the chromosomes condense and they tightly coil, and eventually the nuclear membrane is going to break down and the nucleolus has disappeared. You can't see that anymore. The cell is no longer making proteins, so ribosomal RNA is not being made for the ribosomes because we don't need extra ribosomes. No protein synthesis. During prophase, as I mentioned, you're going to see that the spindle apparatuses shown in this cell have already been formed and the chromosomes are visible. Okay? No nucleolus. Can't see any nucleolus. Then the cell enters metaphase, and all the chromosomes line up at the metaphase plate because of the microtubules that push them there. So the microtubules push the chromosomes towards the metaphase plate, and you can see that right here. The blue chromosomes are lined up in the middle. Anaphase is the next stage, which you can notice because the chromosomes' sister chromatids have been pulled apart. So the centromeres have to split for this to happen. And that's because these spindle fibers now are pulling the sister chromatids towards opposite ends of the cell, away from the metaphase plate. It's pretty majestic, really, that all this happens. And then finally, the last stage is telophase. And I have to mention that telophase, which is part of mitosis, and cytokinesis really coincide. It's almost impossible to tell a cell that is in mitosis's telophase versus cytokinesis because it's kind of happening at the same time. So all the events that occur are that the chromosomes are going to decondense. That means they're going to become less visible over time and go back to this loose chromatin form. You can't see that necessarily happening in these cells yet. A nuclear membrane is going to begin to form. This all takes time. You can see it in the diagram that there's kind of a nuclear membrane forming. Eventually, the nucleolus will reappear, but it will be very faint at first and then get darker. The spindle apparatus and all those microtubules will disappear. And the telltale for me, if I can't see any of the other things, is that a cleavage furrow develops. This cleavage furrow is an indentation and signifies where a cell membrane is going to form to separate the two new daughter cells. So here are some pictures that we'll just go through so that you can learn how to identify. This is important for lab as well, how you can identify cells in different stages. So this cell, I agree there's a lot of extracellular material shown in red. That's just extracellular matrix. And then they're showing you, that's why it's very zoomed in, or I'm sorry, very zoomed out. Now we zoom in to the cell, and this is the whole cell. And you can see that the chromosomes have condensed, so we know right away we're in prophase. And this is just later prophase. The whole cell is shown here. There's no nuclear membrane anymore. There was a itsy bitsy little nucleolus here, but now it's gone. And the chromosomes are completely separated from one another, but yet they're still kind of co-localized where the nucleus was. So I would still call this prophase. This is even later prophase. It's not until the chromosomes are actually on the metaphase plate that we say the cell is in the next stage, metaphase. So I can't tell where the cell membrane is anymore. But this is metaphase because the chromosomes are lined up. Now, the way the chromosomes line up is that the centromere gets put at the metaphase plate. So those chromatids are like arms that kind of flail about. So you'll often see some chromatids winging out like this, okay? But it's still pretty equidistant and lined up in the center. And then finally, we can see in anaphase that these chromosomes, or chromatids, I should say, are being pulled apart one direction versus the other direction in the cell. So this is late anaphase here. You can really tell that the chromatids are separated. And then finally, the last stage is telophase of mitosis, which happens at the same time as cytokinesis. And so here I can tell that this is where a new membrane is going to form. And we've almost got a nucleus forming. Not exactly, I don't see a membrane per se, but kind of. We can tell the chromosomes are all co-localizing. And this is later in cytokinesis because I actually see this faint clear line going down the middle, which signifies the creation of phospholipid membrane or phospholipid bilayer membrane. So one more practice slide or two more practice slides. In this slide, I've got some cells circled and I wanted to go through them with you. In A, give you a minute to think about it, but in cell A, what I notice is that I cannot see any DNA and there's a nucleolus visible. So what stage do you think that would be? It turns out it's interphase. Can't tell what stage that cell is in. Interphase is what we call it. And then in the cell that's labeled B, I can now see chromosomes and no nuclear membrane surrounding them. So this is prophase. And then C, you can see that the chromatids have been separated. So I would call this anaphase. On this particular slide, this picture, microscope slide as well as PowerPoint slide, there are no cells in metaphase. I can't find any cells in metaphase. And then in D, I can tell that what has just happened is telophase and cytokinesis because the cells are small, taking up about the same size as the original cell. And I guess that's the bottom line. You can tell that with a couple of others here. So down here, you can tell that these two cells are recently formed through mitosis and cytokinesis. So let's do one more example, and then you're done with mitosis. So A, I don't see a nucleolus. So my guess for A is prophase. What do you think B is? That's metaphase. And C is anaphase. Now in D, these cells are small. There's two of them. Can't quite exactly make out where the cytoplasm is and the cell membrane, but I guessed here. There's a nuclear membrane that's forming, and I think there's a nucleolus forming right here. So I would put this at telophase / cytokinesis. You could call it either or both. And then in E, I know for sure that these are two cells that just were formed, and I can see in this clear line that, in this clear line, that the cell membrane's forming right there. So those two cells just formed. And then F, I would say the cell is in interphase because there's a clear nucleolus. So that's it for mitosis and the cell cycle. Thank you for your attention.